This video describes prokaryotic replication forks, with a specific focus on the theta model. Theta replication is the main mode in which bacteria, particularly E. coli, replicate. This diagram shows a very simplistic model of theta replication. The circular DNA's two nucleotide strands unwind at the origin of replication, shown here as a yellow rectangle. This is the site in which DNA synthesis is initiated. Unwinding here will generate a replication bubble, and as further unwinding occurs, the formation of a replication fork. This diagram has two, and thus is bidirectional. In the end, two circular DNA molecules will be produced, each containing one old and one new strand, hence why DNA replication is described as semi-conservative. DNA is anti-parallel, so as the two strands unwind, one of the template strands, shown here in black, gets exposed in the 3' to 5' direction. With the help of DNA polymerase, this permits the new strand, shown in green, to be synthesized continuously, making this the leading strand. The other template strand, also shown in black, is exposed in the 5' to 3' direction. As a result, replication occurs for this strand discontinuously and produces the Okazaki fragments. These are short stretches of DNA, shown more clearly in this close-up. These fragments mean that DNA is still elongating in the same 5' to 3' direction on the lagging strand. The remainder of this video will be on deconstructing this at first complicated-looking diagram. Helicase breaks hydrogen bonds to unwind and expose the template DNA. Single-stranded binding proteins, the pink shapes, bind and prevent secondary structures forming. As helicase moves along the strand, the fork will also move in this direction, creating torsional strain ahead. DNA gyrase, the orange oval, is a topoisomerase that relieves this strain. DNA polymerase, however, cannot just start adding nucleotides to a bare template. Instead, it needs an existing 3' hydroxyl group. This is where primase comes in. It synthesizes a short stretch of RNA onto a template, the primer. This provides the needed group. The leading strand needs a singular primer at the 5' end, whereas the lagging strand needs a new primer at the start of each Okazaki, shown in red. The yellow rectangles are DNA polymerase III, and they add new nucleotides to the 3' end of the growing strand. Next to this on the lagging strand in green is DNA polymerase I. Both have similar enzymatic activities, but DNA pol I differs as it has an additional 5' to 3' exonuclease action to allow it to remove the primers and replace them with new nucleotides. This, however, leaves a break in the strand that ligase, the purple rectangle, will correct via the formation of a phosphodiester bond, specifically between the 3' hydroxyl group of one nucleotide and the 5' phosphate group of another. To conclude, for theta replication, as it is bidirectional, termination will occur when the replication forks meet.